before we start, some of the notices that I have to give you, right? So you guys are what we consider as the first intake, right? So you people in this meeting and also in my in that WhatsApp group are considered as my first intake, right? So you are only online people. So what's going to happen is your class is going to continue, right, at the same time at 5 p.m. on Sunday, right? But at the same time, from next week onwards, I'm starting a new class at ISM Nugget, ISM, right? So there are a few institutes that invited me to start, right? So after uh, thinking about each of the institutes, so I decided to settle at ISM, right? So with the time it took to get that decision, to take that decision, so it took some time. So that's why there were some different messages I sent, right? So ISM is the final decision. Mm, that class is at 8:30 Sunday, right? 8:30 a.m. Sunday. So that is going to be from the very beginning. That class is going to start from the very beginning, from your introduction to biology, right? Then in a sort of a rapid way, it's going to catch up to you guys level, right? So it's going to catch up to the place that we are in this class. So I think we have to start extracellular matrix today, right? So up to that level, I'm going to come in about 6 weeks. That's my plan. In about 6 weeks, we are going to come to your level. And once that happens, I'm going to merge the two classes, right? So this 5 p.m. class will start streaming from 8 a.m. on Sunday, 8:30 a.m. on Sunday, right? As a physical, sort of like a video stream of the physical class, right? But that will happen through the Gurutaru online website, right? Even though I'm doing classes at ISM, the online is from the Gurutaru online website because it is, I think, easier for me and also easier for you to have a separate, so that sort of platform because they are doing the tube delivery as well. And that's the other news. Because of some address issues as well as some fees finalizing issues, it took some time to uh, prepare the sort of the posting process of it. So the tutes are in the post right now, but I think nobody has sort of received them, right? Nobody got them, right? Anyone got them? We posted yesterday, but it's Sunday now, so I think, yeah. So probably within the next week you will receive, and that tute has it's in full color, right? It's in full color, and it will have until the end of cell division, right? Everything. Okay. So this tute is, I mean, in another month or so, you are going to get another new tute, right? Because we are almost, we have completed a lot of things in this tute, right? So once all of you have got that tute, next week at 5 p.m. This class, I'm going to have sort of a revision session, right? Revision session next Sunday, right? I'm going to have a revision session. So I will be going through. So once all of you have got the tute in your hands, I'll be doing like a hour session, 1 hour session to complete the tute. Because I think some of you haven't taken printouts and some of you haven't sort of completed everything I have told you to write down like that in the tute. So once all of you have gotten the tute in your hands, I'll be doing a 1 hour, usually maybe a 1 hour session, so that your tute will be complete, right? So that your tute will be complete. That will happen on next Sunday once all of you have gotten your tutes. Okay. Any questions? And also, from next week onwards, I'm going to start a warm-up paper. It will most likely contain a few MCQs, and maybe one structured essay question from what we did the previous week, right? A small warm-up paper. It will take only about 15 minutes to do. And within another 15 minutes, we'll discuss it. And that will sort of, I guess, it will warm you up for the learning process that you are going to start, right? So it's a warm-up paper. And at the end, I'll try to do this, not sure, but the end of the paper, end of the day, there will be a day revision paper. Day revision. Again, another small 15-minute paper, right? That won't be delivered to you. It will be displayed in your, I'll screen share that paper, right? So the day revision and warm-up papers, that will start most likely from next week, all right? So rather than, so we are anyway going to do past paper questions during the class, but also you're going to get these two kinds of papers as well, right? Otherwise, I think your sort of active learning part is a bit missing from me just talking and talking, because I don't give you a written note, because I think that's a waste of time giving you something to write other than what, I mean, giving you a note from what is there in the resource book is actually a waste of time, right? If I'm going to tell you to write down something, that is going to be something additional for what is there in the resource book, right? So because I don't do that, I think some of your active work during the class is not enough. So that's why we are going to do this two kinds of papers at the start of the class, at the end of the class. This will happen in the live class as well as this will happen in the online class as well. Right? Any questions? And also, so for the online people, I am planning that this will be your last class that I share the link to this group, right? So from next month onwards, the link is available in the Gurutaru online website. So you have to join from there, right? Maybe next week also I put the link, but definitely the week after, no more joining from the group. Okay, you have to join from the website. So that is basically it. All the, what do you say, messages, notices I have to give you. Any questions? So next Sunday, we are going to have a 1 hour of tute completion session. Tute completion session, 1 hour. And then we are going to have maybe not a warm-up paper, but a day revision paper, definitely. All right. Yeah. Any questions? That paper will be like an exam. Not really. You can refer the paper. Sorry, you can refer the resource book. You can refer the books. You can refer. It's what I call as, so this warm-up paper and this day revision are what I call as part of your, I mean, I have this sort of 3-step scientific learning thing, right? What are the 3 steps? Understanding, active recall, and systematic repetition. So I sort of consider this warm-up paper as systematic repetition, right? So you learned it last week, then we are learning it. You are reminding this again, right? I'm thinking day revision is part of what? Day revision is part of what step? Active recall, right? Active recall. Get it? Any questions? Do you have any issues from the Gurudhar online website side? Any issues with payments? All right. You have this Prasanga is his name. He's sort of dedicated to my students, right? So he's available not 24 hours, maybe not after 10 p.m. But you can call him anytime if there are issues. He's totally updated, right? Any issue with tutes not receiving, fee payment, and anything that is wrong in the website, you have to tell him, okay? But if you don't understand any subject content, if you have any personal issues, if you have any doubts about what you learned, anything like that, you have my personal number, right? So you can send me a message. Okay. Right. Any questions? So anyone who's going to join the physical class next week? Answer at ISM Logo. It's a free session. Next week, first class is free. Anyone? No? All of you are going to continue online. Okay, all right. All right, where did we stop? We discussed about cell junctions, right? We discussed about cell junctions. You can hear me, right? This thing's working. Okay. We finished the cell junctions. So once the two classes are merged, your class time will most probably be increased to about 3.5 hours, 3 hours, maybe, right? Because this is an online, you don't get a break, right? I don't need to give you a break now. So we usually finish in about 2 hours. So once the physical once the two classes are joined, the live streaming of the physical and the online, the class time will increase to about 3.5 hours. Okay, so right. Then we have to start today with the extracellular matrix, right? Extracellular matrix. So once we have completed the extracellular matrix, that is the end of the cellular basis section, right? Before that, we had to go through that comparison chart, right? So I have conveniently put that chart at the back of your tube, because in this part of your sort of let's say studying stage, this is the chart that you should always try to memorize again and again. You're going to come across charts like this, several charts like this in your syllabus. This comparison between prokaryotic and eukaryotic cells is sort of the first chart that you will encounter like that. There are several other charts like that. There are vitamins that you have to memorize, right? Vitamins that has functions and deficiency symptoms. Then minerals are there. So minerals, there are two parts, as plant minerals and animal minerals, right? Then you have what? Then you have plant growth substances. There's a chart. Plant growth substances. There's a chart of all the functions. Then what else? There's a vitamin chart. Yeah, plant minerals chart is there. Animal minerals chart is there. There are some under evolution. You have some eras, right? Eras and eons, right? So when eras means like when the Earth is evolving, have you heard the Hadean Eon, Archean Eon, Jurassic, Triassic eras, right? Jurassic era, you might have heard definitely, right? So those kind of things, what happens in each era, you have to memorize. So there are a few charts like this in your syllabus, right? Right? So you should, you never can memorize them in one session, and you will always forget, right? You might always forget the facts in those charts. So you have to either make a wallpaper and paste in your room, or I think if you have a sort of a back cover like this, you can sort of always look at it again and again, right? So that is my plan with that. To be honest, what I usually did for the vitamins and minerals, I pasted in my toilet. Okay, I had these charts. I wrote them down with my hand, and I had pasted in the wall of the toilet, so I can see while I'm sitting on the toilet as well as while I'm taking a bath. Right. So every morning, I won't say the duration, right? So every morning I'm studying at least one vitamin or mineral, its function and its deficiency, right? So otherwise, you will never, if you never, I mean, if you don't study it again and again, you will never remember it. So, right. So that is that story. So let's finish extracellular matrix today, and then come to this chart that I ignored earlier. Right. Okay. Okay. Take out your resource books. Sorry. So last week, I think we started from cell walls, right? So quick reminders about the things we learned, right? How many types of cell walls are there in? So in this section, we talk mainly about plant cells, no? But what are the other organisms that have a cell wall? Fungi have a cell wall. Bacteria have a cell wall, right? Fungal cell wall is composed of usually chitin, right? Bacterial cell wall, we say peptidoglycans, right? Is a type of polysaccharide with some proteins. Okay. But here we are mainly talking about plant cell walls, right? Plant cell wall, what is the main composition? Cellulose, hemicellulose, right? And the land that there can be lignin and suberin. And what's the pasting substance in between two plant cell walls? You have pectin, right? So in terms of the time period when these cell walls are produced, right? Who is produced first? The primary cell wall. Then if they are going to get a secondary cell wall, right? Not all plant cells get a secondary cell wall, right? I hope you understood that, right? There are plant cells who live with only a primary cell wall and then they die. But there are cells who get a secondary cell wall and then they die, right? Some cells have only a primary cell wall throughout their life cycle. But some cells get a secondary cell wall and that is the reason for your death. Why is the secondary cell wall is a death sentence? Because it is impermeable to water and other substances, right? Water and water dissolved substances, right? So, what is the location of the primary cell wall? It is just outside the plasma membrane. And what are the features of it? It's permeable. It's usually thinner than the secondary cell wall. It's flexible and composed of cellulose and mainly cellulose and hemicellulose. Secondary cell wall, what is the location? It is between the plasma membrane and the primary cell wall. You have the secondary cell wall. What are the main composition? Lignin mainly, suberin could be also be there. It's a rigid structure, impermeable, right? And then those are the things. Okay. Then if we come to cell junctions, right? I hope you are going through the resource book while I'm reading this, page 38. Okay. Cell junctions, what is the definition? Are structures at which cytoplasms of adjoining cells are joined. Okay. So cell junctions are only found in animal cells, right? Massive MCQ point. Cell junctions can only be found in animal cells. Three types of cell junctions are there. What? Tight junctions, desmosomes or anchor junctions, and gap junctions or communicating junctions. Three types. What is the definition of a tight junction? They connect plasma membranes of adjacent cells with plasma membranes tightly bound together by specific proteins, forming continuous seals. Right? What are the keywords? Plasma membranes are tightly bound, forming continuous seals. Those are the two keywords when you are describing tight junctions. Okay. Main purpose is prevent the leakage of extracellular fluid into intracellular space. Right? It's like supergluing two plasma membranes together. Right? Two plasma membranes are superglued together. The location example is in your skin. I gave you another example where can you find tight junctions? Where else can you find tight junctions other than the skin? intestinal, say, yes, intestinal epithelium. Epithelium is the outer cell layer, all right, to prevent the leakage of whatever you are eating, right? Whatever semi-digested food with enzymes, acids from leaking into your inside your intestine, right? The wall of the intestine, right? Okay. Then desmosomes or anchor junctions are, we can consider they are the strongest type, right? They mechanically connect the cytoskeletons by what sort of filaments? Intermediate filaments, which are the strongest type of filaments as well. But because the cytoskeletons are connected, it is the also the strongest type of cell junction found in muscle tissue, right? Gap junctions and communicating junctions, cytoplasmic channels from one cell to the adjacent cell. They are composed of special membrane proteins surrounding a pore, right? What are the things that can pass through it? Ions, sugars, amino acids. Okay. What are the locations in your cardiac muscle? Heart muscle, that is, we don't consider it heart muscle, it's cardiac muscle. Then animal embryo, right? I told you in animal embryo, when it's just a ball of cells, right? All these cells should properly communicate with each other to decide how they are going to differentiate, how they are going to develop into a complete organism, right? So that is done by this gap junctions, all the communication. Then finally we have plasmodesmata. So do we consider plasmodesmata as a cell junction? Is plasmodesmata a cell junction? No, right? They are just holes in the plasma, sorry, in the cell wall, right? There are holes in the cell wall. It's like I told you, between your neighbor and your house, if you have a blind wall, right? Just a wall separating your two houses. If you drill a hole, if you break that wall down, there's a hole there. That's like your plasmodesmata, right? So the two cells are directly connected and the cytoplasm is shared through that door. The cellular contents are shared. But the biggest thing is you have to remember the cellular membrane, sorry, the plasma membrane also goes through the plasmodesmata into the next cell as well. Plasmodesmata is shared. Sorry, through the plasmodesmata, the plasma membrane is shared. Okay. Right. Then we have to start extracellular matrix. What do you know about the extracellular matrix? So because we just talked about plant cell walls, so we know animal cells don't have a cell wall. But because animal cells only have what outside their cell? They only have a plasma membrane. But we know plasma membrane is a very delicate structure. It is composed of phospholipids, right? Phospholipids are very small. So only that phospholipid bilayer, that is your plasma membrane, is not sufficient to just provide the support given by your cell wall to a plant cell. Similar amount of support is not given to animal cells. Do you understand what I'm saying? Because plant cells are okay. Plant cells get a lot of support from their plant cell wall. But animal cells do not have such a cell wall. But their cells need support. So they have come up with a method to get that support, but in a much more elaborate way, in a much more complicated. Right? Your cell wall is just a layer of cellulose and hemicellulose and other things. What the animal cells did is they developed a much more complex supportive system. Much more complex supportive system. All right. So I'll draw it. No need to draw it because you don't need to be able to draw the extracellular matrix. I'll just screen share. Okay. You don't need to be able to draw the extracellular matrix. I have a good diagram of the extracellular matrix in your tube. I'll screen share the tube, then I can explain. Okay. So can you see my screen? Yes. So in your tunes, I won't be focusing on completion of the tube because we are going to have a session next week about how to complete extracellular matrix. Is the topic. All right. Extracellular matrix of animal cells, the more complete topic. All right. So what can you see in this diagram? This is your plasma membrane, phospholipid bilayer. Can you see? These are the phospholipids. What are these? Just tell me what is the class of molecule, right? So these are phospholipids are lipids, no? What are these? These are proteins, right? Proteins, right? What are they specifically? So you know we have two types of proteins in the plasma membrane, right? I hope you remember, right? If they are embedded in the bilayer, we call them, they are embedded in the bilayer, we call them what? Anyone? There are two types of proteins in the plasma membrane. No. There's a sort of a hint you can get from this diagram as well. It says integrin. Integrin. So what are the proteins that are sort of embedded in the plasma membrane called in the phospholipid bilayer? Integral proteins. Integral proteins, right? But what is the other type? I'll just tell you. What is the other type of protein that are not actually embedded but are loosely bound to the interior? What are they called? Peripheral proteins. No? Peripheral proteins. So you have to start studying now, right? So now your results have come. Now we have completely confirmed that you are doing A-level bio. So now you have to start studying. I told you in the previous 3 months, I didn't tell you to study, right? So now I'm saying just keep in touch with what you're doing. Otherwise, you will have a bit of a workload when it comes to your first term tests, right? So keep in touch with what we are doing in class. So this will be easier once we start these sort of revision papers, right? So peripheral proteins. So you can't see a peripheral protein here, so I'll erase that, right? Integral proteins. But so for an additional point, right? For some, uh, I'll be, I mean, it will impress me if someone can tell me this. This is a special type of integral proteins. These two proteins are two special type of integral proteins. Can anyone tell me? Yes, they are integral proteins, but what is sort of yeah, okay, I got answers. Can someone tell me what is the sort of the special yes, okay. Very good. Very good. Right? These are not just integral proteins, these are what? They traverse or what? They sort of, what is the keyword that you're using? They traverse, right? Or they span the entire membrane. Is a keyword. I'm careful to stick to those keywords because then it's easier for you to remember. What does it say? Uh, yes, some proteins that penetrate all the way through, right? What are they? Penetrate all the way through now. These are special type of integral proteins that we call as what? Transmembrane. transmembrane proteins, right? So none of these are important here, right? So I'm just revising. Okay. So, right. Then it's come to discussing this extracellular matrix. So because as I told you, animal cells do not have a good supportive structure because they don't have a cell wall. What they have is this entire sort of complex that we call as extracellular matrix, right? This is there to provide support. Okay, this is there to provide support. Okay, so what are the components? You can see collagen. Can you see collagen fibers? Collagen fibers. Yes, those are the predominant component of the extracellular matrix. Yes. So collagen, I hope you remember now, collagen is a protein, right? Can anyone tell me what is the protein sort of organization level of protein of collagen? Organization level of protein in collagen. In your tube, I actually specifically put a diagram when we did proteins. What is the organization level of collagen? Collagen protein. Yes, very good. Very good. It's a quaternary structure of protein. Because why? Quaternary structure of proteins are composed of two or more polypeptide chains. So how many polypeptide chains are there in collagen? Three polypeptide chains come together and it twists around each other, forming like a rope-like structure. Right? We usually take three smaller ropes and imagine twisting it to make a bigger rope. Collagen is like that. Collagen is very, very strong. Right? So collagen is the predominant sort of the component, right? It's a protein, right? So collagen fibers you can see. Then there's this among these collagen fibers, we have what we call as a proteoglycan complex, right? Proteoglycan complex. So can you see these sort of wavy? These are like those like hydrilla plants that you can see in ponds. Can you see? Right? So they sort of are woven in and out of these collagen fibers, the proteoglycan complexes. This is the structure of proteoglycan. There's a central polysaccharide, right? There's a central polysaccharide molecule, and it has these sort of branches. And in those branches, you have this protein polypeptide chains. And from those branches, you have sort of leaves like things that are actually small proteins. Sorry, carbohydrates. Sorry. I'll repeat again. There's a long polysaccharide molecule that is sort of like the stem of a proteoglycan. Then from that, you have proteins that are coming out, right? And from those proteins, you have these sort of leaves, small leaves like components that are actually formed of carbohydrate. Okay. So you can label this. We actually call this protein as a co-protein. Co-protein. And we call this, so this is the proteoglycan complex. This entire section is a proteoglycan complex. But if you take like one of these branches, we call it a proteoglycan molecule. Proteoglycan molecule. Right? It's a proteoglycan molecule. So that contains what? A central core protein and carbohydrates, sort of leaves coming out of that core protein. All right. Okay. So we talked about collagen. We have proteoglycan that are sort of connecting or sort of the collagen fibers are embedded in this network of proteoglycan. And the other important thing is, can you see, these collagen fibers are connected to these transmembrane proteins by these small protein fragments that we call as what? Fibronectins. Fibronectins. Okay? Fibronectins. Got it? What are the functions? Mainly same functions of cell membrane, sorry, cell wall. That is what? Forming a protective layer, over the surface of the animal cell. But other than the functions of a cell wall, your extracellular matrix is much more, let's say, much more intelligent and much more useful than a cell wall, because it has the ability to sort of connect animal cells mechanically as well as chemically, right? Mechanically as well as chemically. It's like your houses, adjacent houses are connected by different wires. No? You have electricity coming, you have telephone lines coming. Like that, your houses are sort of interconnected. No? So you can take calls to the next house using your telephone lines, right? Like that, your extracellular matrix has this sort of signaling role, right? Direct signaling role. Okay, that is an additional function of the extracellular matrix. And also, I told you, can you see this collagen are connected to this transmembrane protein, which has a special name called integrin, by this green color protein that is fibronectin. And can you see at the interior of the cell, can you see these filaments? These are parts of your cytoskeleton. These are parts of your cytoskeleton. So what is happening? Your extracellular matrix is connected through these integrins to the interior cytoskeleton, right? So your extracellular matrix and the cytoskeleton actually communicate and are physically connected with each other. So that is going to give the cell a lot of support, right? So the outer collagen is connected to inner what? Inner cytoskeletal elements, mainly microfilaments. And inside you have intermediate filaments, microtubules, right? Parts of your cytoskeleton. That is another feature or another function that we can see in the extracellular matrix. Okay. So that is basically what I have to explain. I'll give a small note, then we can go through the resource as well. Any questions? Any questions? For the new students, I mean, you can join our, you can join our brand new class online stream of our brand new class to start from the very beginning. That is the best thing for you to do, because this class will continue at this sort of pace until the two classes catch up together. All right. So any questions? No, right? Then we can move on to the resource book. There's nothing much here. So even though animal cells lack a cell wall, they have an elaborate extracellular matrix. Main components are what? Glycoproteins and other carbohydrates secreted by the molecule, carbohydrate containing molecules secreted by cells. Then these are MCQ points. What is the most abundant glycoprotein is collagen, right? Then which forms strong fibers outside the cell. Okay. Then collagen is embedded in a network woven out of proteoglycans secreted by cells. Collagen fibers are connected to integral proteins of plasma membrane via fibronectin. So all those things you can see in the diagram, right? Collagen fibers are there. Collagen are embedded in a proteoglycan network, right? Proteoglycan consists of proteoglycan molecules. Collagen is connected to the integral proteins by fibronectin. On the other hand, these integral proteins are connected to your cytoskeleton inside the cell. Okay. So what are the functions? Protective layer over the cell surface, links extracellular matrix and the cytoskeleton, influence cell behavior by involving mechanical and chemical signals. Right? There can be mechanical signals directly coming from those collagen fibers and the proteoglycan. There could be chemical signals coming from the proteoglycan complex. Okay. We can write down a small few points here in this space, right? Write down a few points in this space. Write down. Write down. A proteoglycan molecule, write down. A proteoglycan molecule consists of a coprotein. Proteoglycan molecule consists of a coprotein with consists of a coprotein with coprotein C-O-R-E with carbohydrate chains. Proteoglycan complexes are formed. Proteoglycan complexes are formed. Proteoglycan complexes are formed when many proteoglycan molecules when many proteoglycan molecules when many proteoglycan molecules are attached to a are attached to a are attached to a long polysaccharide are attached to a long polysaccharide. Are attached to a long polysaccharide. Integrins are special type of transmembrane proteins. Integrins are special type of transmembrane proteins that mechanically connect that mechanically connect that mechanically connect extracellular matrix extracellular matrix and the cytoskeleton. Extracellular matrix and the cytoskeleton. Okay. Is there any MCQ that we haven't done in the tute itself? Any MCQ? Yes or no? Before that, let's come to the chart, right? That is in your page. So, any question from the cellular matrix? No, right? Right? No. So, come to the chart in page 25. Differences between prokaryotic and eukaryotic cells. Okay. So, for about now 3 weeks or 4 weeks, all the organelles that we discussed are mainly belongs to the eukaryotic cells. Okay, all the organelles that we discussed are usually eukaryotic cells, eukaryotic organelles that belong to eukaryotic cells, right? But now it is very important that we compare how prokaryotic and eukaryotic cells differ, right? That comparison is what we are going to do. So, actually, in evolution, you will come to see that all the organisms in the world, all the living organisms, we classify into what we call as three domains. Domains. Rajadhani කියලා කියනවා in Sinhala. Domains. Right? Domains. Right? Three domains. Right? As what? Bacteria, domain bacteria, domain archaea, archaea, archaea and eukarya. These are the three domains. Right? So this one you are familiar. Eukaryotic, eukarya. Right? So where is the prokaryotic then? So if there is eukaryotic, where is the word prokaryotic? Why are we not classifying animals as eukaryotic and prokaryotic? Right? These two domains are what we call as prokaryotic. Prokaryotic. Right? So these domains were actually a very recent discovery. Very recent discovery. Right? Domains. Right? I think in 1970s we discovered these domains. Right? Anybody know what is the next sort of the classification under domain? We have kingdoms, right? Under eukarya, we have kingdoms. Kingdom plants we have. Kingdom animalia, animals we have. Kingdom protista we have. Kingdom fungi we have. These are kingdoms, right? So actually, these bacteria and archaea are microorganisms, right? Are microorganisms and they are prokaryotic. They are the prokaryotes. Most of the organisms that you can see in the world belongs to the eukaryotic category. All the plants, all the animals, all the algae and protozoa, and all the fungi, they are all eukaryotic organisms, right? These two that are considered as prokaryotic, they are microorganisms, right? Actually, bacteria, and these are also called as archaebacteria. Archaea are also called as archaebacteria, right? So what is the difference between these bacteria and archaebacteria? What is the difference? Anybody know? You haven't heard the word, right? Archaebacteria? Archaebacteria or archaea? The biggest difference is that these archaebacteria are bacteria that can live in extreme conditions. That can live in extreme conditions. What are extreme conditions? In our world, there are some extreme places. What are these extremes? Extremes are like more than the normal level. What are extremes? High temperature, right? Temperature can be very high or very low. That's an extreme, right? So it can be very, very hot or very, very cold environments are there, right? Then what else? Very highly acidic environments, right? Or basic environment. What else? High salt, salinity, very high salt concentration environments are there, right? Like that. So high temperature, there are bacteria living in volcanoes, right? In volcanic environments, there are bacteria living, right? Then there are bacteria living in hot water springs, right? And also pressure, extremes of pressure, very high pressure. There are bacteria living in very high pressure conditions, such as where? In the very deep sea, there are bacteria living, right? So when scientists explored these extreme bacteria, they came to realize that they are very different from the normal bacteria that we think about, right? So these bacteria that are in these extreme places are actually very different to the normal bacteria that we talk about. That's why we actually classified them separately as archaebacteria. You following me, right? They have so many differences that have enabled them to live in these extreme places, right? Following me up to this point, right? Right, salt marshes, high salt places. What are you can see salt marshes, right? Like what do you say? වගුරු බිම් කියලා කියන්නේ ගොඩක් salinity තියෙන, right? Near your lagoons, near the ocean, you have a very highly salinity, very high places such as salt marshes. Normal bacteria cannot survive because the salt is going to take the water from inside the cell out. It's going to dry the cell because of this high salinity. But this archaebacteria can survive, right? So why am I telling you this? Because at the same time that we consider them both as prokaryotic, but there are massive differences between normal bacteria and archaebacteria. Okay, keep that in mind, right? So come to the chart in your page 26, and sorry, 25. The chart starts at page 25. What is the first feature? What are the examples of organisms? For bacteria, you have, sorry, for prokaryotes, you have the bacteria, archaebacteria. For eukaryotes, you have protists, fungi, animals, plant, right? Those are the main examples that you can take, right? Then cell size, usually your prokaryotic cells are usually very small, 0.5 to 5 micrometers. Micrometers. What is micrometers? 10 to the power minus 6, right? 10 to the power minus 6. Okay. So what is the average size of a eukaryotic cell? 10 micrometers to 100 micrometers, right? Usually then about twice big to maybe 100 times bigger. Usually eukaryotic cells are compared to prokaryotic cells about 100 times bigger, right? Then the form, right? Usually prokaryotic cells stay as single cells, right? So that we call it as unicellular, single cell. Then eukaryotes are usually multicellular. If you take an animal, right, it is composed of millions of cells. The exception is some of your protists and some fungi can exist as single cells. Right? Have you heard amoeba, euglena, paramecium? These are all protists that can exist as uh, unicellular. Right? There are some fungi as well, that can exist as single cells. But even fungi can make a multicellular community. Have you seen like a piece of bread that is having fungus? Puskala That you can see there's like a white color sort of like cotton wool like, right? So that's a colony of fungi. So usually fungi also exist as multicellular. But there are few instances, such as in protists, that there are unicellular eukaryotes. Okay. Then evolutionary origin, right? Anybody know how many billion years ago did life originate on Earth? We think, right? However, if it came from space or it just originated here on Earth by its own, we don't know. මෙහි automatically හැදුනද එහෙම නැත්නම් it came through a volcano, sorry, not a volcano, a meteorite, උල්කාපාතයක්, right? We don't know, right? However, the earliest fossils that we have found that have like that looks like earliest cells are 4.6 billion years old. 4.6 billion years. Okay. Right? 4.6 billion years old. But those cells, even those early cells, we cannot even consider prokaryotic cells. Okay. Even the earliest cells, we cannot even consider it as prokaryotic cells. We actually call them as protocells. 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 They are like not even cells at all. They are just collections of biological molecules. They are just collections of molecules that are sort of look like a cell, right? For the proper, even the proper prokaryotic cells originated about 3.5 billion years. So from that stage to become a prokaryotic cell, it took about what? About 1 billion years. It took about 1 billion years, right? So that's how slow evolution was, like a billion years. Can you imagine? Right? What is the sort of the power of a billion? 10 to the power 9 is million, no? 12. 10 to the power 12, right? Billion. Ah, sorry, I didn't see. Sorry, there was a request to repeat the note. Okay, thank you. All right. So you got it right. So what is the sort of the power of billion? 10 to the power 12, right? I think so. Yeah. So this is how long it even took for this sort of very basic cells to become even prokaryotic cells, right? So what is the evolutionary origin of prokaryotic? 3.5 billion years ago. For the first eukaryotic cells to originate, it took about another how many? So eukaryotic cells originate about 1.8 billion years ago, right? What is the difference? 3.5 to about another 1.8 billion year difference, right? 1.7 billion years kind of difference between the prokaryotic and eukaryotic cell origin. Okay, origin. Then cell division is binary fission, no mitosis or meiosis, right? But in eukaryotes, you can see mitosis, meiosis, or both. So that is the next section that we are going to talk about, right? How cells divide, how one cell becomes two, right? So I'll explain this at the end because today we are going to start cell division now, right? So just keep that for now. I'll explain what is binary fission. It's a very simple form of cells dividing. So binary fission you can see in your prokaryotes. In eukaryotes, you can see two types of cell divisions, either mitosis or meiosis. Genetic material, it doesn't matter you are a prokaryote or eukaryote. What is your genetic material? It's DNA. It's DNA. Universal genetic material is DNA, right? But there are many big differences between the DNA of prokaryotes and eukaryotes, right? Since prokaryotic cells does not have a nucleus. They do not have a nucleus, right? The DNA is just lying there in the cytoplasm, but it has a particular region, right? It's lying in a particular region of the cytoplasm that we call as nucleoid. Nucleoid, right? Very important. Remember that it's called the nucleoid, all right? And the DNA, we consider it as circular DNA. Circular, it is coiled. It's like just tightly packed into a coil that we consider as circular DNA. I'll show you that, right? Circular DNA, what is circular DNA? And it is not associated with histone. So you know what is histone, right? I showed you the histone protein, right? It's a type of protein and it forms this sort of 8-molecule block. 8-molecule block, 8 histones. And around this, you have DNA wrapping. So DNA molecules wrap twice around histones. Okay. So that is the DNA, how genetic material is arranged. I'll show you this right now, otherwise you might forget. So you learn about this, how DNA is packaged and arranged in prokaryotic cells and eukaryotic cells in much more detail in the section that I'm doing for the 2025 students, right? 2025. That is one of your most sort of advanced lessons called molecular biology, right? Molecular biology and recombinant DNA technology. That is your 7th lesson, I think. Yes, 7th. So it's like this. So this is a histone molecule, you can see, right? You can see 8 histone molecules, right? 1, 2, 3, 4, and 1, 2, 3, 4. That's 8 comes together to form this protein complex called composed of histone protein. We actually call it as a nucleosome. Okay, nucleosome. And you can see the DNA wraps around twice. Okay, so then I'll show you what a circular DNA, this sort of circular DNA looks like in prokaryotes, right? I'm explaining this right now because otherwise, if you don't understand, you can't memorize, right? What is even circular DNA? You are going to wonder, right? So without you properly understanding it, you cannot properly memorize, right? So this is what a circular DNA looks like. Can you see? It's a circle, right? That's why we call it circular DNA. But actually, inside the bacterial cell or prokaryotic cell, this doesn't exist as a complete circle. Why? Can you look at this diagram and tell me why this DNA doesn't exist like a whole big circle? 10 to the power 9 is 1 billion. Okay. 10 to the power 6 is. No, but the 10 to the power 9 is million, right? No, 10 to the power 9 is billion, right? Yeah. 6 is million, no? Yeah. So anyway, what I'm asking is, why can't this circular DNA, can you see the big circle, right? Why can't it exist like that inside the prokaryotic cell? Anybody? Just, let's say, just general knowledge. Why can't it just exist as a circular chromosome inside the prokaryotic cell? What's the problem? Can you see the size of it? Yes, exactly. Yes, very good. 350 micrometers is the sort of the diameter, right? But what is the average size of a prokaryotic cell? 0.5 to 0.5 to 5 micrometers, right? So that is about, I don't know, right, many times smaller than this big circular chromosome. What is going to happen is it's going to what is going to happen is like there's a process called prokaryotic DNA packaging, and at the end it is going to pack into a very small structure about 2 micrometers, right? 2 micrometers in diameter. That is again in average prokaryotic cell, right? So there are very small cells. So in them, this is going to be even smaller, right? So this is like a average size. Okay. So can you understand now what is circular DNA? All right. But in your eukaryotes, you have linear DNA, right? Linear DNA. So this is another example of this circular DNA, right? It's a big circle, right? This is a DNA in E. coli. E. coli is a type of bacteria, right? Um, but in our sort of in eukaryotic cells, right? DNA exists as what? DNA exists linearly as linear molecules. I show you how they exist as well, like this, right? So this is a histones. Can you see these are histones? So DNA wraps around them, and there's some several stages of DNA packaging we call, and at the end they form these sort of very compact structures called chromosomes, Varnadeha, right? So in us, in eukaryotic cells, there's a starting point for the DNA and there's an ending point, start point and end point, right? So it's not circular. We call it a linear DNA model. You're clear? Linear. Okay, got it right? Yeah, right. Okay. Then what is the next classification? Um, Genetic material we talked about, type of ribosomes, right? Ribosome. So you know there are two types of ribosomes. I told you, right? What are the two types? As 70s and 80s. 70s ribosomes and 80s ribosomes, right? So what is this number S mean? What is this number S mean? It refers to the size of the ribosome. It refers to the size of the ribosome, right? So actually your 70s, whatever, 70s and 80s are both composed of two parts, right? Two parts. What are the two parts? There's a large subunit and a small subunit, right? I hope you remember. There's a large part and a small part, right? Right. So the upper part, the large part is like this. There's a lower small part like this. Large subunit and a small subunit. Okay. So in a 70s ribosome, this actually the large part we call it as 50S, and the small part is actually I remember 30S, I think. So yes, the two numbers don't add up to 70, but if we individually consider their size, it's 50 and 30. We don't need that, okay? This is 70. And if you take like a 80S, if I can remember, it's like a 60S plus 40S. The large subunit is 60 and the small subunit is 40. Right? See, this S is so it's a unit, right? Hold on. I just learned this in our microbiology. I just learned what is this S. Even I didn't know what is this S when I did my A-levels. So I'm not properly studying microbiology. That seems just learned about this. Hold on. So in your third year, you come across a subject called microbiology. There you have to learn about all these structures again and again. Yes. In this S refers to, yeah, it's called the sedimentation coefficient, right? It's a molecular biological technique, right? Sedimentation coefficient. S means sedimentation coefficient, right? So you are 70s because they are smaller, they have a smaller sedimentation coefficient. You are 80s because they are bigger, they have a higher sedimentation coefficient, right? Just you don't need these numbers. What are their individual sizes? You don't need, right? Just know prokaryotic cells have 70s. Eukaryotic cells have eukaryotic cells have what? 70s or 80s? What does eukaryotic cells have? They have both, right? They have both. Where do eukaryotic cells have 70s? I told you there's a theory, right? Endosymbiont theory, right? Early in evolution, eukaryotic cells ate prokaryotic cells. I hope you remember. Go back and read about the endosymbiont theory, right? Some time ago, maybe in this gap from 3.5 billion of prokaryotic cells to 1.8 billion of eukaryotic, maybe a eukaryotic cell ate a prokaryotic cells, right? I told you, right? So that's how we got what? Inside eukaryotic cells, we think that's how we got chloroplast and mitochondria. When a eukaryotic cell ate a photosynthetic prokaryotic cell, that prokaryotic cell inside became a chloroplast. That's a theory that we have. And if that eukaryotic cell ate a, let's say, a respirating prokaryotic cell that is producing energy, that became mitochondria. So because your mitochondria and chloroplast are sort of like prokaryotes inside a eukaryote, they still have 70s ribosomes. They still have 70s ribosomes, right? So where do you have 80s ribosomes in eukaryotic cells? Attached to, in two places, attached to the rough endoplasmic reticulum, or just as small groups lying everywhere in the cytoplasm. Okay? That is the ribosome differences. Any questions? No, right? Then we can come to organelles, right? Very, very important classific, comparison. Prokaryotic cells do not have organelles. Prokaryotic cells do not have organelles, right? Only what? What do you have? You only have subcellular components, all right? Um, put a star mark and write down if you want. Put a star mark and write down in your resource book, maybe with a pencil if you don't like writing in the resource book. Write down a subcellular component, a subcellular component bound by a membrane, bound by a membrane, a subcellular component bound by a membrane, the subcellular component bound by a membrane becomes an organelle, becomes an organelle. Subcellular component bound by a membrane becomes an organelle. Okay, got it right? So remember, prokaryotic cells do not have any organelles. They only have subcellular components, right? They only have subcellular components. What are an example of a subcellular component that you can see in prokaryotic cells? You can have ribosomes, 70S ribosomes, right? Then if you take the eukaryotic, there are so many membrane-bound organelles. Even among those, we can have two categories as single membrane-bound organelles and double membrane-bound organelles, right? Please highlight in your page 26. You have what are the double membrane-bound organelles? Nucleus, mitochondria, chloroplast, double membrane. Then single membrane-bound organelles, lysosomes, and vacuole. They have only one membrane. Very important. Okay. Then coming back to prokaryotic, internal membranes are scarce. Scarce, not scarces, it's scarce. Scarce means what? Adui කියන එක, rare. Right? වැඩිය දකින්න නැහැ. If present, usually associated with respiration, photosynthesis, or nitrogen fixation. Right. Nitrogen fixation is a very important thing that is done by prokaryotic cells. N2. N2 is what? Your atmospheric nitrogen. N2 gas, nitrogen gas. This nitrogen gas is very unusable. You can't use it for anything, useless. Why is that? Chemistry. You are doing these days, structure and bonding, right? That's the second lesson. Structure and bonding? I can't remember. Yeah. So in that you have to draw these Lewis structures, right? What is the structure of nitrogen? How do you draw nitrogen? N2. How many bonds do you have between two nitrogen atoms? 3, no? And there's a lone pair here. Are you doing cellular, sorry, structure and bonding in chemistry? Yes. Right? So, how many bonds do you have between two nitrogens? 3 bonds. Very, very difficult to separate the two nitrogens because 3 bonds, right? So, this N2 in our atmosphere is useless. We can't use it for anything. Therefore, prokaryotes come into play. They have specific enzymes that can break this nitrogen, take the atoms, and convert it into much more useful forms, such as nitrate, NO3 minus, and ammonium, NH4 plus. Have you heard of this? These are free radicals, right? These radicals, or I mean, they are molecules. These things can be used by other organisms, such as your plants, fungi. They can use this nitrates and ammoniums. These are useless. They are useful. This is useless. But only prokaryotes have the ability to convert this useless N2 gas into these useful forms of nitrogen. Do you understand? All right. So to do that, some of these nitrogen fixing prokaryotes have specialized membranes inside the cell. Got it? Right? That is the story about organelles. See, I told you, without doing these organelles, right? Without discussing what is endosymbiont theory, right? What is without discussing the structure of ribosomes, without discussing the structure of chloroplast, mitochondria, cell membrane, we cannot discuss this chart, right? That's why I kept it to do after we have discussed all the organelles and subcellular structures, right? Then if I started discussing this chart before talking anything about these organelles, then it will be useless for both of us. I'll be wasting my time and you will be, I mean, The understanding portion will be less. You get it? Right? Then we can come to cell walls. So we just talked about cell walls. So we are now experts in cell walls. Right? But here we are going to compare the cell walls between prokaryotic and eukaryotic. We only talked about eukaryotic cells, mainly in plant cell walls. Right? So your bacteria and these archibacteria have different structural components in their cell walls. Very, very important. Right? Your bacteria have cell walls composed of peptidoglycan. Peptidoglycan. All right? Peptidoglycan. Your archibacteria have cell wall composed of polysaccharides and proteins. Polysaccharide and proteins. Why is that important? Why is that important? Yes, why is that important? Archaea has polysaccharide and proteins. Because why? I told you, your archibacteria needs to survive where? In extreme conditions, in extreme temperatures, extreme pressure, extreme salt concentration, acid concentration, right? So just a normal cell wall composed of peptidoglycan is not going to make them survive those extreme conditions. But archaea, because they have polysaccharide and proteins in their cell wall, they are very well protected. They are very well protected. That is the advantage of the cell wall in archaebacteria. Okay, right? So you know, when talking about eukaryotic cell walls, plants and fungi have cell walls, right? So we talked about plant cell walls, right? Mainly composed of cellulose, hemicellulose, and fungal cell walls have mainly chitin. Okay. So under protist, we have algae, right? Algae. Have you seen algae? That's they are floating everywhere in the ocean, right? This sort of green color algae are there, brown color algae are there. We use algae to make what? Right? Algae. We extract something called alginic acid that we use to make ice cream. Iser. I don't know if we use it anymore. Algae, right? Then we get something called agar. Agar we used in to culture bacteria. Bacteria culture. It's like a jelly, right? Agar, right? That is extracted from algae cell walls, right? So protista have also some cell walls. Okay. Then flagella. We talked a lot about the flagella, right? So if we talked about the prokaryotic flagella, they are simple. They are not composed of microtubules, right? So we talked about eukaryotic flagella. It is composed of microtubules arranged in 9+2 structure, right? Microtubules are arranged 9 doublets around 2 single microtubules, right? So that is the flagellum. Flagellum is attached to where? What is the structure that is there when where the flagellum attaches? Anyone? What do we call the place where the flagellum attaches? Yeah, seaweed is mostly algae. What we consider as seaweed are types of algae. Right, I asked, what do you call? Yes, very good. The place where the flagellum attaches to the cell membrane is called the basal body. Basal body has a slightly different structure that is called 9+0. It has 9 triplets of microtubules without any central microtubules. Please go back and read flagellum structure, very important. And the next important thing is that entire flagellum is covered by the plasma membrane as well. It is covered by the cell membrane. So we consider it as a intracellular. It belongs to the inside of the cell, right? Because it is covered by the plasma membrane. Everything that is inside the plasma membrane is intracellular, right? But that is not the case in prokaryotic cells. They don't have microtubules. Microtubules nothing. Anyway, they don't have 9+2 structure. No? So they don't have microtubules, therefore they don't have 9+2 structure. And the primitive flagellum they have, mainly composed of proteins, it is also not covered by their plasma membrane as well. So it is extracellular. Extracellular. Okay, got it. Then respiration, mostly by internal membrane foldings, right? Prokaryotic cells do not have a mitochondria, right? Because why? A prokaryotic cell became a mitochondria according to the theory, right? That's a prokaryotic cell became a mitochondria, right? So in mitochondria, can you remember the inner membrane has these foldings that is inside coming, I mean, inner foldings. What do you call those inner foldings? So now this is a revision class, right? What do you call the inner membrane foldings in mitochondria? Cristae. Very good. So that's what they're talking about here, right? What do they say under prokaryotes? Internal membrane foldings, right? So that's like a mitochondria. So a prokaryotic cell is like a mitochondria, right? So the inner membrane has folded. That's what performs respiration in prokaryotic cells, right? So that is similar to the structure of a mitochondria in eukaryotic cells. Then photosynthesis, no chloroplast, because why? They became the chloroplast, right? Prokaryotic cell sometime later became the chloroplast. Takes place on membranes that show no stacking. Stacking means what? I hope you remember. Inside the chloroplast, you have this inner membrane system called what? Inner membrane system called what? Thylakoid membrane that are sort of stacked, right? Stacked like a sort of sandbags, flat sandbags. Yeah, no, not sandbags, like rice bags. Yeah, halgoni goda halal, halgoni, right? Thylakoid membrane. These are like stacked halgoni, right? Rice bags, right? These membranes are called thylakoids, and this like a godak, like halgoni godak, like a stack of these membranes are called what? Anyone? Anyone? What do you call this stack? Very good. It's a granum, plural grana, right? These two are grana. These grana are connected by what? Grana are connected by what? Five, four, three, two. Intergranal lamella. Very good, right? So this is a stack of thylakoid that we call as granum. So that stacking you can't see, right? So even though we say, okay, a prokaryotic photosynthetic cell became a chloroplast, but that original prokaryotic cell didn't have this sort of stacked membranes. That sort of evolved after they came inside the eukaryotic cell to save space, to have more membrane packed inside the chloroplast. They underwent this stacking to form the granum. Okay. Understood. No stacking. There's an inner membrane system. That's it. So you have chloroplast, which have usually stacked, right, into granum. Then finally, nitrogen fixation. I told you none of the eukaryotic cells have the ability to fix this useless N2 gas into useful forms, such as nitrates, ammonium, right, like that. That is the comparison. So write down a homework. We are going to write an essay. Write down a homework. Briefly compare. Briefly compare. Briefly compare structural differences. Briefly compare structural differences between. Briefly compare structural differences between prokaryotic cells and eukaryotic cells. Briefly compare structural differences between prokaryotic cells and eukaryotic cells. Did I give any homework previous week? Any essays to write? I forget and you don't remind. It is happening. Did I give any homework? Previously, any essays? No? Okay. Okay. Right. So this is your homework using this chart. So did I tell you how to study using essay-based studying? Right. So what you do is you study this chart, taking however long you want. Right. Highlight everything, memorize, close the book, and write this essay. Differences between prokaryotic and eukaryotic. Write essays, write an essay. Don't use points, okay? Don't use bullets, right? मैं point दा लिया है, bullets दा लिया है essays, right? You don't write using in point form. You write complete sentences. It's an essay. Rachana prashna. You have to write an essay, right? Properly break paragraphs. What I recommend is break paragraphs at these different features, right? Break a paragraph for cell division, break a paragraph for DNA, break a paragraph when you're comparing ribosomes, break a paragraph when you're comparing organelles, cell walls, like that. Then the examiner knows that you have a very thorough memory of how to properly compare. That is going to do two things. Not, it has two sides of benefits. From your side, it is going to make it easy for you to not miss anything because you are breaking paragraphs at different features. You get what I mean? Break paragraphs at the features. Write about organelles, break a paragraph. Start by cell walls, like that. Then once you have written the essay, then you can just quickly scan through it and just check, have I written about organelles or cell walls? Have I written about their DNA? Like that, you can just quickly scan and see, yes, the essay is complete. That's your side of benefit. And from the examiner's side, those are again your benefits now. The examiner is easier to read your essay. For the examiner, it's easier to read your essay. So that gives like a good impression, right? Even though if you have messy handwriting, if your essay is organized properly, chapters are properly separated, right? That is going to give a good impression and probably increase your marks, maybe by 1 point or 2 points, because you know two people are going to mark your essay, right? There are two examiners. But it doesn't matter. Both examiners are going to be sort of satisfied with your essay, even though you have such unclear or messy handwriting. But if you have properly organized your answer like that, that is going to be a big benefit. You get it? Because once you are in grade 13, there is no time to sort of, let's say, correct your essay writing technique, right? Correct it right now. When you're writing every essay, write it in a proper order, write it with proper organization. Doesn't matter if your handwriting is not, I mean, it just has to be clear. It is just has to be legible, readable. That's all. right? One of my friends, what he did was he has such bad handwriting. He started to write essays in massive letters. And then he left a rule, like like leaving a rule after one after the other because his handwriting is so messy. But he got an 8, 3 years actually, right? So whatever the strategy you need to make your answer easily readable, right? That's what you need. So that's the homework. Right. Before we move on, yeah, doctor's handwriting are messy. Yeah. So several reasons for that, right? That is exact. I mean, I think it's 95% of medical faculties fault, right? And 5% of doctors sort of carelessness. Not carelessness. They don't pay attention to write neatly. So exact. So if you, I mean, in our exams, in the university exam that we face, all right, there is no marking scheme. For example, for many subjects, there is no marking scheme, fixed set of points that you are given marks. For example, in your first 2 years, if you ever get into medicine, you have a subject called anatomy, right? Anatomy. That is studying the structure of your human body, right? So for anatomy, there are so many textbooks, right? There's an Indian one called Chaurasya, this one, right? That's what I used because it has some good diagrams, right? That's what I used, right? There's this one. So that's what the average student uses, right? Then there's another one called, have you heard Grey's Anatomy? There's a TV show in it. Grey's Anatomy. There's a student version of, that's an actual book, Grey's Anatomy, right? So there are people who are studying that book. Then there's another book called Last, there's Cunningham's, whatever, right? So depending on what textbook you are using, you know different things about anatomy. So for example, they ask you to write about what do you say, layers of your neck, right? What are the layers that you have from the center to the outside? Depending what book you are studying, they can have different descriptions, right? So when you are writing the paper, different people are going to write different things. So therefore, there's no fixed marking scheme for a subject like anatomy, right? So what you have to do is to go to the exam and you have to write everything you know. You have to write everything you know about a particular. So if they ask about the neck, you have to write everything you know. And a single essay question is given 20 minutes. And in your context, what you know, if you get a essay from proteins, you know there's a limited amount of points that you have to target, right? Limited amount of points that you have to write. But in our exam, what we have is there is no limit to the knowledge. There is no limit to what you can write, right? So in order to pass, you have to write as much as possible. And then if you want to get a distinction, right? They are given distinctions in a subject, right? If you have more than 75, it's called a distinction. So if you are a distinction student, you have to write so many things. You have to draw diagrams. So I think that is one of the places where we get our messy handwriting because we have to write very fast. And the second place is when we go to the hospital, we have to take notes from patients. We have to do what we call as a history taking, right? We have to ask the patient why they are here, right? What medications they are on, what previous surgeries, what previous diseases they have had, their family history, what is their problem, right? We have to analyze the problem, what are the symptoms they are having. Then we have to examine the patient. We have to write everything down very fast, right? We have to write everything down very fast. Usually I use my tab, right? And those handwriting only I can read, right? Because we have to usually then when the consultant comes, we have to present that history to the consultant. So only us, I mean, only we should be able to read that just because it's just a note. So that is another place that we are getting our messy handwriting. So it's like that. That's why it's a sort of a toxic cycle. Right. Then, are there any questions that we didn't do from cellular basis? Any MCQ? Yes or no? 4, 3, 4. Oh yeah. There's a few, no? Yeah, the screen share. Did we do this question? This one? This number 4? So I didn't number these these questions. One thing I forgot to number. Then the other thing is I wanted to show you what where in the paper you get these questions, what number. So usually it's question number 3 or 4, 3 or 4 or 2. Very rarely, there's a separate set of questions from 40 to 50, right? It's a different type of question from question number 40 to 50. So there you can ask from everywhere, but usually your lessons, the questions are made in your lesson order, right? So definitely your cellular basis will be within the first 10 questions, right? Expect within the first 5 actually, right? So that's why I just left the numbers like that without correctly numbering. So have we done this question? This is number 4. Okay, do it. 30 seconds, not take 1 minute. Okay, it's the answer. Okay, there's a variety. So I just need the combination. Okay, A and B, B and D, B and D, A and B, B and D. What about the others? There's 11 people in the meeting. I'm not reading out your names, so don't. And also, you're sending me messages to the main chat, right? Is there anyone sending messages to me privately? No, right? Yeah, yeah. Please send to the main chat. All right, let's discuss presence of presence of plasma. So what are they asking? Common to prokaryotic and eukaryotic cells. Presence of plasma membrane. B, presence of 70S. Occurrence of mitosis. D, presence of subcellular components suspended in cytosol. Common. Yes. Both of them have plasma membrane. A, yes. Presence of 70S ribosomes. Yes. It's a common feature. Occurrence of mitosis, no. You do not have mitosis cell division or meiosis cell division in prokaryotic cells. Presence of subcellular components suspended in cytosol, D, yes. Subcellular components suspended in cytosol. Hmm. So definitely, yeah, you have subcellular components. You have ribosomes in both prokaryotic and eukaryotic cells. So D is okay. Both have plasma membrane, yes. Both have 70S ribosomes, yes. I think this is one of the questions where they gave all. What do you think? I can't remember the year. Yeah, this I think there's no answer. It has to be ABD, you know? What do you think? It has to be ABD, right? Why not D? Of course, prokaryotes have plasma membrane. Of course. See, all we went through this, right? What are the all cells? What are the features in all cells? They have a plasma membrane. They have a cytosol. They have subcellular components. They have DNA as genetic material. And ribosomes are found in all cells. These are the features that you can see in all cells. All cells. Right? I didn't go through this. But anyway, now you know, right? All cells have a plasma membrane. All cells have a jelly-like substance inside called cytosol. Inside in that you have subcellular components. Right? Subcellular components are there in all prokaryotic or eukaryotic cells, but organelles are found only in eukaryotic cells. They have DNA as genetic material, yes. Ribosomes are found in all cells. So yes, the proper answer should be A, B, D, no? Yeah, it's A, B, D is the answer. There's no answer like that. Yeah, okay. What else did we not do? Did we do this one? There's no number. I don't know why there's no number. Right, this question. Try that. Did you do it? This question? No, right? Yeah, you can. It's an all. Yeah, yeah.
I think that year it's an all for any answer they have given marks. I'll check and see with the marking scheme. I can't remember the what year that is. Did we do this question that is in the screen? Yes or no? No, right? Try. What do you mean no membrane bound? In which question? In the previous one. Okay, this one in the screen. Have you done it? Okay, send me the answers for this. Send me answers. Okay. The one in the red box that's in the screen that I sort of circle with the red pen. You can see the screen, right? Yeah. Where do you go to? Physics and chemistry? People who are from around Colombo. Where do you go for physics and chemistry? Okay, I'm getting answers. Two. Okay. Right. Structure and function. Nucleolus ribosome production. Remember the nucleolus, right? Nucleolus is located inside the nucleus. It's when you observe under the electron microscope, you can see it's a darkly stained area.
because it has some synthesizing function. Not usually, there's not much DNA. There's a synthesizing function. It produces rRNA. Ribosomal RNA is produced there. rRNA. So rRNA is one of the biggest, one of the main components of ribosomes. Is rRNA. That's true, nucleolus. Glyoxysomes, uh, photorespiration in plants. No, photorespiration, what is involved in photorespiration? Your mitochondria and peroxisomes, right? So we can write down a review. Mitochondria and peroxisomes are involved in photorespiration. We'll learn what is photorespiration in very soon when you're doing photosynthesis. Mitochondria and peroxisomes are involved in photorespiration, not glyoxysomes. Okay, right. Cytoskeleton determines the shape of the cell. It provides support, yes. Is there a function specifically saying that cytoskeleton gives shape, gives a shape? Yeah, maintain shape. Yeah. Then vacuoles store soluble. They are asking, I think, the incorrect one, right? There's a part of the question missing. There's a part of the question missing, right? I printed this question like this, no? Ah, I might need to correct that. I think they're asking the incorrect statement. They're asking the incorrect statement. I'll check this and tell you, okay? They're asking the incorrect one, right? We are getting correct ones. No? So number 2 is the only incorrect one. Vacuoles. Number 4, vacuoles store soluble substances needed for cellular activities. Yes, right? In the tonoplast, right? Sorry, not the tonoplast. Tonoplast is the membrane. In the cell sap of the vacuole, you have soluble substances such as pigments, sugars, right? Golgi complex produce lysosomes. Golgi produce lysosomes. It's true, right? Golgi produce lysosomes. That's true. So they are asking the incorrect one. So please correct it. What is the incorrect statement? Is number 2. So photorespiration and glyoxysomes. Glyoxysomes are involved in what? Converting fatty acids into sugars. That is the function. It's a special type of peroxisome. So photorespiration is, again, if you're talking about photorespiration, it's a wasteful process, right? During photosynthesis, this wasteful process happens when when the light reaching the chloroplast in a photosynthetic cell, usually a plant, is very high. When the sunlight is very high and also the temperature is very high. There's a wasteful process called photorespiration happening that produces a toxic component, right? It produces a toxic chemical called 2-phosphoglycolate that is in your syllabus. That has to be processed. That has to be broken down. 2-phosphoglycolate has to be broken down. That requires enzymes in the peroxisome and mitochondria as well. Mitochondria is necessary to sort of not for the breakdown, but to further remove it. Okay. Main role is done by peroxisome. Got it? Any doubts? Can I repeat the review? What did I tell? mitochondria and peroxisomes are involved in photorespiration of plants. That's the review, right? Yeah. Okay.
Any doubts? Any questions? Okay. 3 and 4 in mitochondria in cytoskeleton not done.
These two are not done. Hmm. I think in this question number 3, you require a bit of additional knowledge. Intermembrane space. Pyruvate is converted to acetyl CoA. What are they asking here? So are they asking the correct one? Right. So the statement is now, so statement number 4, you don't know anything about. No? You don't know what is pyruvate. You don't know what is acetyl coenzyme A. All right. Yeah. Leave that question. Right. You can try question. This question. This number question number 4. You can try that. Try that in the cytoskeleton. Yeah. Okay. Done. Okay. All right. We can discuss, right? What's the answer?
In the cytoskeleton. Um,
They're asking, usually they're asking for the correct statement, right? Let's discuss. In the cytoskeleton, microtubules are formed by actin. Yes, actin microtubules. Actin microtubules. Okay. Then keratin is not present. That's wrong, right? Because your intermediate filaments can be composed of keratin, right? Am I right? So keratin can be present. Microtubules are involved in movement of organelles. Now this is a little bit, even though this is a new syllabus paper, I told you your sort of resource book underwent a few changes, especially in this cytoskeleton part. So some of the features, sorry, the functions of the cytoskeletal elements are missing. So usually movement of organelles, it is mm, microtubules, is it given as a, let's keep that. Microfilaments are involved in movement of, sorry. The number one is microtubules, or very sorry, right? Microtubules are so this is microfilaments, right? Microtubules are composed of what? Tubulin, tubulin, very sorry, right? So that's wrong. Microtubules are not composed of actin. Write down a small review. Write down a small review. Microtubules are composed of tubulin monomer. Microtubules are composed of tubulin monomer, while microtubules are composed of tubulin monomer, while microfilaments are composed of actin. Microfilaments are composed of actin. Yeah, my mistake. Right? Microfilaments are composed of actin. Yeah. Keratin is usually, so number two, keratin is not present, that is false. Microtubules is involved in the movement of organelles, that should be the correct statement, but I think it's missing from your resource. Let's see. Microfilaments are involved in movement of chromosomes during cell division. I specifically told you this, right? During cell division, your DNA will be pulled into opposite directions because you are going to make two cells from one cell. So your DNA needs to be divided. So that DNA will be taken to opposite sides by microtubules. You can just cut this, you can underline these microfilaments and just say it's not microfilaments, it's microtubules. Right? Statement number 5, intermediate filaments provide channels to secrete material from the cell. No, right? Intermediate filaments are basically involved in providing structural strength and support, right? And also intermediate filaments are involved in what? Forming anchor junctions, desmosomes. So what are they talking about here? These channels that are communicating junctions or gap junctions, right? So that's sort of there to confuse you. So the correct statement is microtubules. They are they are involved in movement of organelles. Yes, that is from your old syllabus, not old syllabus, old resource book. There's a whole sort of paragraph missing. I think I have my old resource book when I did my A-levels. Yes, organelle movements is a function of microtubules කියලා දීලා තියෙනවා in the sort of the old version of the resource book. Okay, you can write down there. Organelle movements is a function of microtubules.
Okay. Great. Any questions? Can I review the fifth answer? Intermediate provide channels provide filaments provide channels to secrete materials. Um, we can write down. Intermediate filaments are involved in forming. Write down a small review for the fifth statement in that question. Intermediate filaments are involved in forming. Intermediate filaments are involved in forming anchor junctions. Anchor junctions. Intermediate filaments are involved in forming anchor junctions. Not gap junctions. Not gap junctions. Which form, not gap junctions, which form, not gap junctions, which form cytoplasmic channels from one cell to the another, from cytoplasmic channels from one cell to the adjacent cell. Not gap junctions, which provide cytoplasmic channels from one cell to the adjacent cell. You got it right? Don't ask me to repeat it. Okay. So can someone put it in the chat for anyone who didn't manage to write it? Okay. So can you see? So if you have a small memory, there are some sort of a filament, there's some sort of a junction that is formed by intermediate filaments, and there's a type of junction that is sort of involved in this sort of making a pore between cells. If you have like a light memory, you're going to get confused there, right? If you sometimes you may forget what are intermediate filaments. So then you sometimes get confused. Yeah, there's this sort of filament that is forming a junction. Then there's a junction that is sort of involved in connecting two cells together by forming a pore. Then you're going to get confused and sometimes mark the wrong answer, right? That's why it's in biology. It's a bit it's a cruel sometimes, because if you have a light memory, it's not enough. You have to have a precise memory. I'm saying this because when you're studying from this point onwards, you have to start studying precisely, right? Pay attention to the small details. Otherwise, even the easiest MCQs, such as these very first, right? Such as these very first MCQs in the paper, there's 50 MCQs to come. We are still in this third and fourth kind of MCQs, right? So these are what we consider easy MCQs, right? So you cannot, let's say, you cannot get these MCQs wrong. Otherwise, going to an A is going to be very difficult. To expect an A, you need to have at least above 40. But even though if you have above 40, if you mess up the second paper, so better to have at least 44 to 43 to 45 MCQs correct if you want to sort of ensure you are A pass, right? So MCQ paper is very important. Every single MCQ gets you 1 mark out of 100. That is very high value, right? So if you write a massive essay, maybe 2 pages, massive essay, that is going to get you how many maximum marks out of 100? A massive essay is going to get you how many marks out of 100? Maximum. Essay with 50 points is going to get you how many marks out of 100?
Even the island first usually can't get a complete essay 50 out of 50 points, right? Even if you write a complete essay, the maximum amount of marks you're going to get is 7.5 marks out of 100. That is the maximum. But a single MCQ is going to give you 1 mark out of 100. That's how important MCQs are, right? So, from this, starting from this stage in your A level life, pay attention to the small details, understand properly, take it in. I mean, then it's very unlikely that you are going to miss anything if you have understood properly, right? If you know what is the function of a desmosome or anchor junction, then you know to do that function you require the strongest fiber, strongest filament. That is the intermediate filament, right? So like that, always have a logic behind every theory. Other than you won't be confused, right? I think there's no time for us to get into the introduction of cellular basis. Cell division today. So that is going to be a topic for next week, all right? So we can end there today. If you are willing to join the physical class, you are invited to come to the free class next Sunday at ISM. And this online class will happen as usual at 5 p.m. on Sunday. And you can share the sort of the post that I sent to the group if you have friends that are looking for a bio class, or just anyone who is already going to a bio class, but anyone can walk in and sit down because it's a free session, right? So that's all. Any questions from your side? Any questions? Right? No, no. Then we can finish. Okay. Thank you for joining. 2026. Not sure. I'm not sure for 2026 whether I will start, right? Because doing a small scale class in an institute is not very, let's say, profitable, right? Better to do the online. Let's see if I find some time to do some marketing for 2026, maybe I'll start. Okay. Right. Okay. Then we can stop for today. Thank you for joining. Good night. Okay. Sure, you too. Okay. Study well. Okay. Great.